we often have the question on, what is the difference between iPSCs and animal models? I really see them as two complementary tools. They're very different and they address very different questions. You need an in vivo model, you need an animal model because of the complexity of the in vivo, right? Where you can understand that obviously, again, a disease is not just the brain, but is part of, especially in MS, where you have immune cells migrating from the periphery. So you need to have the big picture. On the other side, though, and you imagine when your car is broken and you want to open the engine, you want to look piece by piece. And so you want to have more reductionist models in which you exclude any confounding factors. And this is something you cannot do in an in vivo model. In addition, instead, you can select one specific type. And so, for example, you can just take all the brain cells, you can recreate in a dish just the glial cells and the neurons, and eliminate any confounding effect that is due from inflammation, from migrating immune cells, and just look at cell autonomous effect. So this will allow to address, again, very different questions. And on top of that, you also have cells that have the genetic background of the individuals. This is a major point for all neurodegenerative diseases. We know that many neurodegenerative diseases are a huge burden in our society, and they are increasing as the society as we age. And often you will hear say we are not mouse, right? And so the mouse cannot really depict or reflect the entire complexity genetics of the individuals, and in particular the heterogeneity of the individuals. And so MS is in particular a disease in which every person has really his own journey, his own severity, his own response to the drugs, and this is largely due to all these genetic components. We're now talking about SNPs variants, so these tiny polymorphisms that are hundreds of them, and collectively create a balance of what is your risk and what is, again, the severity of your disease. And again, we don't know many of them. We're now discovering, we know a lot of them that are in genes, so in the sequences that codify for proteins, but there are a lot of also other regulatory sequences that we still largely unexplored, and that probably the balance of all these create what is your unique journey to the disease. The IPS modeling gives you now the opportunity to create in a dish, for example, oligodendrocytes, which are the vulnerable cells of neurons which die in the disease, and looking for drugs and test it directly which patients could be the best responders or the weakest responders. So this is a way of largely improving the preclinical phase by already generating information on how to design the clinical trial. So the idea is to use this IPS modeling really robustly in preclinical phase. So we expand that and we go on with only drugs that work at this stage. And so hopefully reducing the failure in clinical phase, which is the most expensive one, which is the largest one, and where often, over 90% of neuro drugs typically fail.